Hello, welcome back. Welcome. I don't know. Okay, so let's talk about phospholipids. Before which, we're gonna talk about this video. Not sponsored by anyone. Me, it's just me and yeah, my books. But whatever. Not butthurt at all. Whatever. Yeah, says what, and then proceeds to say whatever like 15 times in a row. Acha. So this video is made for self-revision purposes only. That being said, my finals is in 1 week, so I do not want to read the book again. I will have to write a research paper, another assignment, a viva, which I'm guessing like it's an oral test, along with a couple of other things. I mean, I chose Rasputin for my research paper. Like, girl, get that. Whatever. So because of all of that and, you know, ADHD, I do not want to read the book again. Hence, I made videos for myself to explain myself again right before the exam, revise, because I'm very stubborn. I don't listen to anyone who ain't me. So this video will have a lot of examples just to help me remember, which are probably not scientific. So if this is not you, Ashvia, do your own research. And also, Ashvia, please do your own freaking research. But let's get started. Phospholipids. We know what phospholipids are, right? This is how you spell it, and I am sure of the spelling. Phospholipids. Okay? So as you can see, phospholipids. What does this mean? Just gonna copy this whole thing, right? That being said, phospholipid is like this. So this is my carbon chain. This is like a little backbone type of stuff. I'm trying my best to control my language. I want to go PG. CH2O CO and then O P. Okay. And then CH2 N. This is what my book says, but I'm not sure if this is the one that I remember like learning initially, but this is what a phospholipid looks like. Okay, just to be clear, you can see it from here that this is like an R group. Okay, it's like hydrocarbons. What are hydrocarbons? Hydrocarbons are hydrophobic. That means this is the lipid part. Okay, and this would be a triacylglycerol, but instead of like the last we got a phosphate thingy. Okay, this is what makes it hydrophilic. This is the hydrophilic end, while this is the hydrophobic end. Remember soaps? Remember my video, Tyler Durden 101? That thing. Okay, so just like soaps, phospholipids also form that little. This is probably not how you draw it, but like just bear with me. This is the hydrophobic tail, while this is the hydrophilic head. Now, I used to be like super confused with this one thing, okay? Like when I first learned about phospholipids, is the tail hydrophobic or is like the head hydrophobic? How do I know? Now, there's this little thing, okay, in my culture, of course. This is called cultural promotion, okay? People, get your diaries. I mean, clearly you won't get your diaries for learning chemistry. You will get your diary because I'm giving lessons on culture promotion. So, in my culture, okay? The feet is considered something offensive. Okay, so let's just say you punch someone, okay? Like you punch someone really hard, okay? And then you punched another, okay, so you punched person A really hard, okay? But you punched person B like slightly. It's not even gonna hurt you. Type. Both of them, same closeness, whatever. You know who's gonna be more offended? Person B. Feet is considered, for some reason, like this very disrespectful thing, like, you know, kicking and all that crap. Feet is related to tail. Okay, keep that in mind. Head is considered to be like it's a very respectful place. Okay, I mean the irony being that you literally use your feet to walk, and without being able to walk, you're kind of considered to be disabled, right? So despite the importance of feet, they consider feet to be disrespectful, which is honestly diabolical in my opinion. Feet is related to tail. Okay, so okay, now let's go to the sidebar where we're going to talk. So in the sidebar, let's talk about hydrophobic and hydrophilic. Hydrophilic is water loving, hydrophobic is water hating. So when you like someone, okay, you cannot get them out of your head. Hydrophilic head. When you dislike someone, okay, they're on your tail. Okay, I feel like I didn't have to bring my culture into this, but whatever. When you really don't like someone, they're kind of like clutching onto your tail. Oh my god, I hope you see this video, you little dip. But whatever. Tail, right? So feet is considered a bad thing. It's like hate, water hating. And so like feet is related to tail, so hydrophobic tail. I hope that makes sense, because I just thought out loud for you. I don't even know if half my videos make sense. I just think it out loud. So this is the whole thing with phospholipids, right? So that being said, they form like bilayer. In our cell membranes, they form bilayer. Okay, so like this is your cell membrane. Like this is the wall of your cell membrane. Like this whole thing together is your cell membrane. Get it? You get it. You kind of really don't have an option here. So you get it. So phospholipids are of generally two types according to my book. Sue my book, not me. Sue my book. Okay, so they are considered to be of two types. One is glycerophospholipids. Okay, this wasn't so hard to pronounce. Glycerophospholipids, right? So we're going to learn about glycerophospholipids and phingosins. I think glycerophospholipids, and then the other one we have is sphingomyelins. Okay, I keep on thinking that it's sphingomycin, but it's actually sphingomyelin. Now remember myelin sheath? I feel like this is a really good way to remember. Myelin sheath, it's like an insulating layer on top of your neurons or axons. I'm not sure. Axons, I think. Okay, because electrical message, the nerve gives off electrical message, and since our body is like 70% water, and water is like a conducting material, semiconducting material, I think. So so the message from our brain to our motor neurons, like on my hand or whatever, it doesn't just like dissociate, get lost or something. That's why axon, which carries the information, I'm guessing, I could be wrong, is covered in myelin sheath, like an insulating layer, right, to create a barrier between our bodies, water, and the neuron, I suppose. So yeah, sphingomyelin. That's how you can possibly remember it, because I do remember myelin sheath very nicely. We're gonna draw this glycerophospholipids. Okay, so I'll draw a few and then we'll come back. Okay, before I get into whatever the hell I've drawn, let's talk about phosphoric acid. Okay, so just to give you a little overview, this is a phosphoric acid. This is a phosphoric acid. Alright? Now, this is phosphoric acid monoester. As you can see, 2OH here and then the ester here, so phosphoric acid monoester. Then we have phosphoric acid diester. Like this whole thing is the name, by the way. OR here, ester here. Phosphoric acid triester, so OR here, OR here, OR here. The one thing that is constant is the double bond between O, oxygen, and phosphor. Keep that in mind and compare that to the carboxylic ester. So as you can see, this is carboxylic, carbon, phosphor. Just have to be clear on that. Okay, this is important if you want to draw phospholipids. Now let's talk about glycerophospholipids. right? So glycerophospholipids, there are a few structures which are kind of like a little pain in you know where to just remember, but it's easy once you really figure it out. Like I drew those on my board, like on my board, and then I just kind of left it there to just like rot for a couple of days. So you know whenever I wake up, I'd see that. So we have phosphatidic acid. I'm gonna draw phosphatidic acid. Good. So there you go. First, this is the backbone for all the glycerophospholipids. So you're gonna draw with me in real time because this is helpful. Okay, C O C O. These are some like you know these structures, glycerophosphates, like phosphatidic acid, which is glycerophospholipid. It's phospholipid structure, right? Phospholipids are in cell membrane. Cell membrane is like a structural component, and structure. Think of architecture. Architecture, they make pretty stuff. So these stuff, they have to be pretty. And sometimes, like as far as I remember, one of the studies once I saw on YouTube, I'm not sure, that symmetric faces are generally considered to be pretty among our toxic beauty standards. Very rare, but very pretty. That's why think of this as symmetric, okay? Then we have O. And then we got CH2, CH. This is where the symmetry kind of breaks. And now here we're gonna have a little bit. Now this thing, yada yada yada, yada yada yada. They have to be the same number. They can range from carbon 12 to carbon 20. Okay, not mentioned here explicitly. Then we have another CH2. Then we got O. Remember those phosphoric acid? So we're just gonna phosphoric acid the crap out of this. O. Whenever you put O here like that, you gotta minus this whole thing. This is your phosphatidic acid. Remember, this right here is your R group. This is your phosphatidic acid. This is your phosphatidic acid. This is your phosphatidic acid. Now keep this constant. This is like a backbone type of stuff. Okay, the same thing that we did for amino acid. You know, we had a backbone and then we just kept on just erasing, drawing. Same thing with steroids. So it's like that. We're not gonna erase. We're just gonna put. Okay, we're also gonna erase a little. So now phosphatidic acid. What is phosphatidylchlorine? Oh my god. Tidylchlorine. Phosphatidylchlorine. So okay, I feel very blind now. Phosphatidylchlorine is when I'm gonna just erase this because this is just gonna create confusion. The O here starts reacting. With what? So now I'm just gonna go back to my skeletal structure. CH2, N plus. Okay, so the moment you see plus, so N has 3 hands, right? The moment you see plus, what does that say? That means it has one more hand than it should have. So that means it has 4 hands. So we already have 1 hand here. Just put it like this, and this. That's it. These things, I'm not gonna, these are CH3, like methyl groups, but I'm not gonna write it because, I mean, for skeletal structure, you really don't need to write it. So this is it. This is your phosphatidylchlorine. Okay, the one way to remember this, phosphatidylchlorine, like the spelling of this, everything else is gonna be the same, by the way. The spelling of this is clonin, Clorox. Clorox is used for cleaning. What else is used for, like, associated with cleaning? Not this dirty rag, but a tidy room. So phosphatidylchlorine, a very bad example. That was a really bad example. Even I wouldn't use it. Then we have phosphatidylethanolamine. What the hell is that? Phosphatidylethanolamine. Phosphatidylethanolamine. Oh my god. Phosphatidyethanolamine. 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 Ethanol is used for sanitizer. Yeah. So, I guess you could see that. I don't know. Okay, I don't know. Oh, this one's fairly easy though. 1. Okay, 2. So we're just gonna have one mountain and one revert mountain. And then we're just gonna have NH3 plus. This is phosphatidyethanolamine. Okay, so I guess this does make sense. Ether means 2 carbons, so CH2, CH2. Amine means NH3, I believe. So yeah, I guess it does make sense. It does make sense. Phosphatidyethanolamine. This makes sense. This makes sense. Now we have phosphatidylserine. Phosphatidylserine. For that, I really want to use purple, but my purple marker ain't working because I really associate sulfur with like purple. I really like that. Phosphatidylserine. Let's do pink. I haven't done pink in such a long time. Phosphatidylserine. So phosphatidylserine, right? Phosphatidylserine. So we have O, and then we got CH2. So one mountain. Okay, another mountain. We have O here. We have, okay. This mountain, we're still not done with this one. Huh. It does look like serine. I mean, not serine, but I mean, I forgot what serine looks like. But it does look like an amino acid, and I think serine is an amino acid, right? Yeah, says the biochem major. So this is it for all the glycerophospholipids. Wait, hold on. Phosphatidylserine. So we had phosphatidic acid, phosphatidylclonine, phosphatidethanolamine, phosphatidylserine. I got that right. Okay, then there was another category. So phospholipids are 2 types: glycerophospholipids and sphingomyelins. So glycerophospholipids have phosphatidic acid, phosphatidylclonine, phosphatidethanolamine, phosphatidylserine. And then we have sphingomycin. Sorry, sphingomyelins are of 2 types: sphingosine and sphingomyelin. I'll get back to you. There we have it. Sphingomyelin are of 2 types. First is sphingomyelin, and then sphingosine. Now, let's draw these structures. Let's learn side by side. So first we're gonna do the side chain first. Okay, so CH3. Oh no, wait, hold on. CH3, CH2, 12. And we're just gonna one mountain, and then we're just gonna go 2 mountain, right? So we're gonna have a double bond right here, if I'm not wrong. Yes, sir. Okay. I don't have to do the 3D structure. OH, OH, NH, 2, and then we're gonna complete this thing. So CH2, OH. This is my sphingosine. Alright? This is my sphingosine. Now you want to keep a few things constant for this. So I'm just gonna redraw this here. Like, just gonna diminish the size again. So it's like easier for me. Let's see how much I remember. This is a fun game. Let's see how much I remember. We're gonna use the blurring method. And I'm gonna try not to see. CH3, CH2, 12, right? We're gonna make 1 mountain and then 2 mountain. And then here we're gonna stop here. The OH is gonna stop here. The NH2 is gonna stop here. The OH is gonna stop here, and there was a double bond right here. Did I do it right? Did I do it right? I think I did it right. Yeah. So we're just gonna draw this whole thing again. So CH3, CH2, 12 of these, and we're gonna draw one mountain, two mountain. We have OH, double bond right here, OH right here, and H2 right here. What is this? This is sphingosine. Now we want to make it sphingomyelin. The sphingomyelin. I feel like this is your first red flag. The name itself is broader. I should have drawn it here. Oh my god. CH3, CH2, 12, 1, 2. This was it, right? Yeah, I think this was this was it. This is sphingosine, for the love of God, this is sphingosine. We're gonna change a few things. That being said, oh, this is N. This was gonna be NH2. So N, we have O. Of course, there's gonna be an H, NH, O, and then we have CH2. Okay, and then CH2 here, which could be 12 to 15, or like 12 to 15 till range, and then CH3. And then this one's also going to change. Now this is a little more complicated. This part is going to be CH2O. Oh, remember those phosphoric acid? Yes, we're going to use that knowledge again. So O, P, O. We have another O. We have another O. Of course we do. CH2, CH2. 1, 2. CH2, CH2. N plus. Hey, I feel like we've drawn this before. 1, 2, 3. I feel like we've drawn this before. This is sphingomyelin. Where did we draw this before? This whole N thing. Ah, phosphatidylchlonine. We drew this before. Like this part. Okay, so phosphatidylchlonine. So this is it. This is how you draw all of it. Oh damn. Best of luck for you. Finals.